It's a great pleasure to welcome Matt Agler from the University of Jena, junior group leader there, to our interview series. Matt, fantastic that you find some time to share your thoughts with us. Matt just gave a seminar in our CRC and is working on plant microbiome interactions in many different aspects. This is a hot field, this is an emerging field, this is a competitive field, and Matt, so you got your PhD at Cornell, and then you moved as a postdoc to Germany, to the Institute of Plant Breeding in Cologne. How did you get to that, in that field, or what attracted you to work on this subject? Can you remember? Does it go back? You always wanted to understand how a plant functions, or how did it all started?
I had kind of an unusual path, I guess, to microbial ecology and plant microbe interactions, especially. I studied as an undergraduate, actually, agricultural and biological engineering. Mm-hmm. And there, for my bachelor's research project, I worked on nutrient cycling, and I had a specialization in microbiology, and I was interested in the role of microbial nutrient cycling there. Mm-hmm. And these questions about microbial ecology and interactions really interested me. So when I did my PhD, I stayed in biological engineering, actually, but I was working with anaerobic microbial communities that convert complex substrates into chemicals, be that methane or acids or whatever useful chemicals, and where the idea was to kind of understand the ecology and use modeling in a way that we can learn to steer these systems to generate certain products. Mm-hmm. And what I realized through all that, I think, is that none of these kind of processes work until microorganisms are coupled and interacting with one another. And so how that happens, like on a fundamental level, really fascinates me. And I decided that I had to do something more basic in this direction of microbial ecology. Mm-hmm. And when I came to Germany, actually, my wife started a junior professor position in Aachen, and I didn't have a position yet. And I was looking around. And I visited different labs and different topics, and I met Eric Kamen in Cologne, mm-hmm. who was working on this question of albugo, and he had this picture of, so albugo is a pathogen of Arabidopsis, and he had this picture of an Arabidopsis leaf stamped on media with albugo and without, and the one with albugo is just covered in all sorts of diversity that grew on the plate then. Yeah, from my background in agricultural related fields and so on, that just hit me like this is a place where there's a lot of relevance for the kind of skills I have. Mm-hmm. And so I come at it from a really different direction, and my time in Cologne was really a baptism with fire on plant microbe interactions. Uh-huh. I guess. Yeah. But for me, it was always about the microbial interactions, and the plant I saw as a system to study that in. Yeah, with a lot of relevance, I guess. Yeah, it's fantastic. Yeah, people come from different points, realizing, wow, that's it, and that's what intrigues me. May I ask you to briefly, in 2-3 sentences, summarize your major research finding in the last few years? You have a wonderful manuscript or paper in bioRxiv, and can you just summarize for our audience very briefly what are the major observations which you made recently as group leader in Jena? Well, we observed generally that leaf microbiota they're not random. They seem to be structured by some force. And we know from previous work that microbial interactions play an important role. So my group looks at how the leaf as an environment shapes that. And what we're finding, for example, is that the leaf defense chemistry determines whether colonization early in a plant's life proceeds stochastically or less so. And we think that this, in turn, via priority effects and so on, and microbial interactions, leads to specific changes in the way that leaves recruit microbial communities. So that would sum it up on a really broad level.
Yeah. Thank you so much. How important were in the field, in your field, and are novel technologies to be able to dig deeper into the problem you are interested in?
I would say really, really critical. I mean, at a really superficial level, maybe a lot of the things that we see and are seeing, a lot of things have been known for a long time. That, colonization of roots can affect susceptibility of disease and so on. And the way that pathogens molecularly interact with plants. But to have now sequencing technologies, for example, and the resources to ask about diversity among pathogens, or about what happens in a community when different organisms are together, would have been really impossible to do previously, because you just physically can't do all of that in a culture dependent way. And there's not always a really clear phenotype to look at. So you need these technologies that allow you to look at broader swaths of diversity to understand those questions. I think it's really critical.
Yeah. And I guess it goes not only with the sequencing, but also with novel visualization technologies, metabolic insights, etc. Yeah. I mean, you're interested in how the plant functions, and you are particularly focusing on the microbial interactions there. But there are billions of plant species available, and you are very much sticking to Arabidopsis. So, of course, this is a model. How important is that to really go with a model, or could the novel technologies just actually allow you just to take any plant of interest living in an interesting environment somewhere? I mean, how important is the model system?
Well, I think it's super important to, of course, be able to pull things apart at the molecular level, right? To understand where do these chemicals come from and things like that. We do some work in other plant species, non-model plants. For example, though, we work in Flavaria. That's a genus that has both C3, plants that do both C3 and C4 photosynthesis. So there's a system where you have closely related plants with very different leaf physiology that might be important, for example, for microbial nutrition. Mm-hmm. And so I think there are really good systems out there to look into other plants. I mean, models are super important. You're right about that, on both the microbe side and the plant side. Mm-hmm. But I think it's also good when people look beyond that as well, to ask whether those answers generalize into other systems, right?
Mm-hmm. Yeah. Let's move a little bit with our conversation. And I mean, looking at your career, it's outstanding. You just, in 2012, you got your PhD at Cornell, and then you moved as a postdoc to Germany. Now you're a group leader. You're doing very independent, very creative own work. For many, the young people here in our center, you're, of course, among the role models. What do you think are factors important for making a successful career? I mean, what was important, and what do you think? What do you want to tell the junior generation? What is really important if you want to stay in this academic sciences?
Yeah, it's a little bit of a difficult question to answer, because, of course, there's more people who would like to stay in there than there are jobs. That's just the reality of it. And most people will find something else eventually. And for me, though, I think a few things. So one is not giving up. I think this is, frankly, it's critical. It has been in my case, for example. So for example, jumping into the plant field for me was a huge challenge, and one I really underestimated when I took that step. And it took time, and I had to give myself that time and accept that, I'm not going to be immediately the top expert there. And so one has to also be humble about those things and know where the limits of your knowledge are. I think that people see right away when that's not the case, and it's important to know those limits as well. But not giving up, having the right mentors or finding mentors is critical. People who will give you honest feedback and help to guide you as you make career decisions. I think this has been for me really important. And people who are paying attention to how things are going for you, so they can give you good feedback. So I've been lucky. When I came here, I got involved right away in the School for Microbial Communication and the Max Planck Research School, and then came Microverse. And I'm a plant person. Jena also doesn't have a huge plant community. So that got me in there, probably, to be perfectly honest. And that has made so many things possible that wouldn't have otherwise been possible, those support systems. So building that up is totally critical, especially when you start as a group leader, for example. And so what I guess if I sum that up, luck is an important part of it as well. There's no question about it.
Yeah, thank you. Probably also unpredictability. You never know. Yes, and that's very nice. Thank you so much. So, I mean, you are telling us how important the people around you were and mentors, etc. Now you are in a very lively place in Jena, and the question is, how important are people outside your immediate discipline? I mean, how important is that thinking beyond the borders, the disciplines, interdisciplinarity, cross-disciplinarity? Does that play a role at the moment in your thinking and in your research, or you rather stay focused where you are and in your discipline?
It plays a really important role. So Jena is a place, I'm sure it's the same where you are with your CRC, but Jena is a place where people tend to be really open to collaborations, and we have really diverse people here. So Jena is really strong in optics and these kind of things. We're really strong in chemical ecology, and we've dived into collaborations on these things head first, and that's, like I said, it increases the capacity of what my group's able to do. We're of course limited as a small group, and we can look a lot broader. So before I came here, I wasn't, for example, looking into the chemical diversity of Arabidopsis because I just didn't have the capacity really to do that. And via collaborations and working with people outside the field, it makes it possible.
Yeah. Is that a challenge for the junior people you get in your lab, for the PhD students and for the young postdocs? I mean, that they all of a sudden have now, well, we go now to the seminar, to the chemistry or to the chemical ecology institute. Is that they have to get used to different languages, I guess. Is that a challenge?
I'm sure it is. I think it's also good. Sure, it's a challenge because, like you said, you have to learn that language. You have to learn how to interpret a different way of describing science, I guess. And but I think it's really good because it also helps students to think outside the box a little bit, to look at things from different perspectives. Yeah, you're right. We have seminars all over the place and many different topics, like you're working. So hearing seminars of someone who's working on human immunity, like you said, there is a lot of overlap, but the language is completely different. And so, and also learning to speak to people like that, to helps to communicate the science better. So it's a big challenge, but I think everybody benefits from it, in my opinion.
Yeah. Now you changed institution several times. You went from Cornell to Cologne, and from Cologne to Jena, and you're still early in your career. What do you expect from an institution in terms of support for a junior group leader? What should be there, and what is necessary? What is missing? What were criteria for choosing a certain institution?
I think as a junior group leader, one is at a stage where you're surrounded by people, but at the same time, very often, I hear it also from other junior group leaders, and I know from myself, one also has constant, like everybody when they start something new, constant imposter syndrome. Is this the right thing? I don't know, when you walk in the door, how do you even get started, kind of thing. And so I think really critical is that, like we talked about earlier, that support system, connecting with other junior group leaders. So there's nothing that can stand in place of that, because that's where you get the most experience from, I think. And then, like I said earlier, that system of mentoring. I think if it would be really good if more places formalized mentoring, so that there's a system in place to make sure that people don't fall through the cracks on things like that.
Yeah. Thank you so much. You mentioned, I think, answering one of the questions in your seminar. Of course, we all went now through a year and a half of difficult times and completely novel challenges. How did you cope with the pandemic in terms of, you're in a sensitive phase of your career, and you are just eager to go on, and now comes this stupid pandemic and all the hygiene and the social distancing rules. How did you cope with that?
It was a big challenge. But for everybody, right? It was learning instantly a completely new way of doing everything. And whenever things first shut down, I think we were at a stage where my first PhD students they should have been thinking about how to wrap things up, which last experiments to do and stuff like that. And then they were out of the lab for a while. And that's aggravating and frustrating for everybody. And I think how we coped with it was in part to deal with it as a group. We met a lot during those times, and we met also just socially, even probably more than usual online, just to keep contact and to keep a sense that we're a community, we're there and moving things forward. And it was challenging at times to move things forward. And so one had to also be understanding, I think, about people's personal situations, and everybody going in and out of quarantine all the time, and stuff like that. Yeah, but it was difficult.
Were there things, experiences which you decided to keep going and not giving up anymore if there's no necessity anymore? Were there some aspects to that?
You mean of things that we decided to give up because of the...
Yes, we were forced to have a completely different life, and we communicate different, etc. Are there some aspects which you think they were not so bad at all, and we keep them going?
Sure, I think a lot of aspects of the digitalization are excellent.
Mm-hmm.
I mean, I no longer in Jena run around from here to there constantly to attend every meeting, right? And this is, if you're careful about not making a meeting every 5 minutes, that can actually save you a crazy big amount of time. That's valuable. Being able to be here today we wouldn't have thought of that, I think, a year and a half ago, right? So this is also great.
Yeah, absolutely. I think everybody makes similar experiences. You are now super active in the field, and you are also shaping the field to some extent on the plant microbiome colonization research. We talked about probiotics and prebiotics and from the plant side, agriculture. Where do you think, what's the perspective of this type of research in future agriculture? I mean, in terms of, will that have any impact? Will that change? Are there new ways of dealing with fertilization and getting better crops or more fertile soils, etc.? Will there be impact in, let's say, in 5 years?
I think so. So you already see today, even on a commercial scale, all of the companies are getting into it. They coat their seeds now with microorganisms that should do this or that. It's out there already. The question is right now the effectiveness, I think. What's the effect size of those things? Is it really economical and so on to do that? And I think that our realization that, for you guys, that meta-organisms exist, right? And that none of these parts, the plant, the pathogen, the rest of the microbiota are functioning on their own. They are all connected, and as we work on that and in these systems try to figure out where those connections come from, I think that's what will give us the power to really implement these seed coatings, for example, reliably, where we can start to understand what do I need to have there to make it stable, to make sure that the environment's not going to mess up what I want to do with the plants. And I think it's about these connections between the organisms. How do plants, rely on their microbiota to adapt to the environment, and why are pathogens, in our case, why are pathogens present on the plants all the time, and there's not always disease. So there's really important questions that I think will have a lot to do with these meta-organisms and or the balance there, to understand that. And I think there's a lot of potential. We have a lot of work to do. 5 years is maybe short to have really major impacts, but it's not far around the corner, I think.
That's a wonderful word for the ending. It's not far around the corner. Thank you so much, Matt Agler from Jena, for sharing your personal views on some of the aspects which are happening in our daily work life. Thank you so much for coming, and thanks and very best wishes for the future.
Thank you. Thanks a lot for having me.
Thank you.